Hey everyone. What I thought I would do today, instead of starting to talk about generalized linear models, which is what I promised you I was going to do, is instead kind of treat this as like a quasi in-class type situation where we can kind of go through a mixed model, the development of a mixed model together, so that way when you're doing your homework, it'll be a little bit more intuitive and easy to do, and it won't have all of the extra stuff that you tend to get when I'm just blathering on about how the models work, et cetera, et cetera. So this will be just much more of a utilitarian exercise. And so I've given you all some data that we'll play with today, and basically what it comes down to is we are interested in the effect of snail herbivory and the application of neonicotinoid pesticides on the amount of glucose available in leaves. So it's just one of many measures of plant quality, and it's actually a good measure of plant quality from the perspective of the plant, because that's what the plant is doing. When it's making air, it's making glucose and then turning it into complex carbohydrates. And so I'm going to just throw up, not that kind of throw up, let's see here, make myself small. And let's just look really quickly at what the experimental design actually looks like, and you'll see why it's appropriate here for our mixed models. And so what we have is we're going to have an experimental design where we either have neonicotinoids or no neonicotinoids applied, and then there's going to either be snails or snails are going to be excluded, which is actually a pretty simple thing to do experimentally, because all you have to do is put little copper rings around the plant, and snails don't like to walk over copper. So here we have what would, classically this would be what we would call a factorial experimental design. The difference is that we've done this design over 4 different fields. So we have plots A, B, C, and D. These we can think of as being drawn from a possible distribution of plots, meaning that we're going to treat these as experimental blocks, and what we're going to probably want to do is account for that in our model, right? And so now we're into the land of thinking about mixed models. Further, this experiment was done over the course of 3 years. And so this sounds very much like a repeated measures design, or you could just think of them as temporal blocks. So there's both a spatial block, which has to do with what plot you're in, as well as these temporal blocks. And these are the data that we're going to want to use to develop a single inclusive model for predicting the amount of glucose. And so again, if we go back here, this is the model that we would imagine setting up, where glucose is some function of snails, some function of neonics, and then some function of snails by neonics. We would want to include that interaction term to make sure that we can trust the hypothesis testing, and then also, if necessary, estimate a parameter that talks about non-additive components of the model. And so if we look at our model here, what we see is that essentially what we've done is we've replicated this experiment 12 times, 4 times each year, and each year we did it across these same four plots. Okay. And so now we know where we're at with the data, and now our challenge is to develop a model to predict the amount of glucose. So again, that simple linear model, except we're going to want to think a little bit about the error structure. And so a good habit to get into when you're doing this kind of thing, here are our data. So right there is leaf glucose, we got neonics, controller treatment, we got plot, we got 4 different plots, we have snail, exclude or don't exclude, and we have time, 2015, 2016, and 2017. So when you have data like this, and this is a pretty good sized piece of data. There's 580 rows, right? So there's a lot of information there. It's a lot of times a good idea to just take a look at it, make sure that it all makes sense and that you understand how the experiment is set up, especially if you're not the one who did the experiment, but if you're trying to help somebody else think about their analyses. So we could start by using this function called table. And I'm just going to say table neonics by snail. So it's going to be these categorical columns, and by creating that table, it's going to tell us how many replicates are in each. So there's 133 replicates where snails were excluded and no neonics were added. There's 148 where snails were excluded and neonics were added, so on and so forth. And we can make that table a little bit bigger. We could add an extra page to it if you want to think about it that way. And so here's the replication across plots. So in total, in plot A, that's the level of replication that we have. In plot B, that's the level of replication we have, so on and so forth. And then I can also add in here the time component. So let's go ahead and add the time component. Now we're getting down to the finest grain that we can get with these particular data. So in 2015, in plot A, this is what our level of replication was. In 2015, in plot B, this is what our level of replication was. And the replication is not going to be exact across all fields, across all years, and that's entirely believable because invariably, you know, a plant dies, or, you know, drop a test tube on the floor in the lab and you just lose specimens. There's a lot of different reasons why things would fail. So we wouldn't necessarily be surprised that it's an unbalanced design. Okay, so we have an idea of what we think we want the model to look like. So I'm going to go ahead and I'm going to just load in the Elmer test library, which again will do the mixed models for us, but it also has the option of having that lowercase ANOVA function that can deal with Elmer objects, which will do it more of the traditional old school kind of way of doing analysis of variance on mixed models. And what I'm going to start with is what I'm going to call a big fat model, because Jim likes to start with big fat models. So let's go ahead and I'm going to cut and paste this big old fat model. Come on, you fat model, you. There it is. So I'm going to put the interaction and the main effects of our fixed effects, whether or not neonics were added and whether or not snails were excluded or not. But then I'm going to really hyperinflate and overparameterize. I know already I'm going to get yelled at by R for doing this, but it's a computer, so I can bug it as much as I want. I'm not only going to let each plot have its own intercept and each time period have its own intercept, but I'm also going to explore the possibility that each plot can have its own intercept as a function of what neonic treatment it had, as well as what snail treatment it had, and then with time, I'm going to explore models where they have their own intercept and slope for the presence or absence of neonics or the presence or absence of snails. So this would be a very fat model. And unless you have an incredibly giant data set and really interesting error structure, invariably if you run this, you're going to get yelled at. And there's our getting yelled at. Boundary fit singular, it's telling us it really doesn't like anything that we've been doing there. You can go ahead and take a look, and we can understand why that might be there, right? Here's that perfect correlation. And what this perfect correlation means is that at the plot level, when it was trying to estimate a separate slope given what neonic treatment it was in, it could not independently arrive at a solution for the slope and the intercept. Instead, it made something that was perfectly correlated. And so, again, the rule of thumb is that you really would like to not put terms into your model that the computer is unable to independently estimate. And so we already know that this model is probably not going to be the model that we want because we're getting yelled at for it being overparameterized. But we can use that step function in Elmer test if we were so inclined. You could also explore a couple of different models. That's always a good idea too. But step allows us to be a little bit lazier. It's not step Elmer test though. Okay, so I'm going to get all these, what are you doing to me? Boundary errors, negative eigenvalues, everything going bad. But what it did eventually wind up on is this model down here. So it's a model that includes neonics, it's a model that includes the presence or absence of snails. It has the interaction term, that's what this little colon here is indicating. And then it also has the error structure, where it's allowing each plot to have its own intercept, which we're going to want anyways. because we know that those plots are not independent of one another and we need to account for that, but it's also allowing for a model where not only do different times have their own intercept, but they also have their own slope based upon what the snail treatment is. So a little bit more of a complex error structure than what we've played with in the lectures in the past. And so now we have a model that we're interested in exploring. And so I'm going to go ahead and cut and paste that model. Let's take a look at that model. I've kind of written up and used the syntax that's more familiar with us, where I've put this interaction term here by just putting the asterisk between our fixed effects. Let me turn off Wi-Fi so nobody else emails me about other stuff I'm supposed to be doing, but I won't. So I have my fixed effects in there and allowing for the interaction, and then I have I'm allowing each plot to have its own intercept, as well as each time period to have its own intercept, and snail to have its own slope, the presence or absence of snail. So let's go ahead and take a look at our model after we've run that. Oh, I probably should run that. And now we can look at our model. And now you can see that it is indeed able to independently estimate the slope based upon snail exclusion, yes or no, in regards to time. And plot is just simply there as something we just want to partition out. So the error structure is a little bit more complicated temporally. And then, of course, we have our parameter estimates that we might feel good about. One thing I did forget to mention very early, it's when you read in things, especially when you're dealing with things like time, where you've put them into the computer as the date or the year, 2014 or whatever, it's really important to make sure that R knows that that is supposed to be a factor and not a number. Because it will treat it very differently if it thinks it's a continuous thing. What we want it to think of is 2015 being the same as if we had typed in the words 2015. So I'm going to just really quickly make sure that I didn't screw that up. I'm pretty sure I didn't. Spoiler alert, this is the second time I've recorded this. The first time I recorded it, the microphone was screwed up on my computer. So you can imagine how thrilled I was. Okay, so what am I doing? Oh, right. I'm going to look at the structure of DAT glucose. And here we have time. Even though they're numbers, R is treating them as factors, which means that R does not know that 2005 is a number. It could have been anything. You could have named it the year of the rabbit, the year of the ox, whatever you wanted to name it. Wouldn't have mattered whatsoever. Okay, but I digress. So now we have our model. And again, we understand why that random slope is valid and the computer's okay with that. And we have our estimates of the parameters in the model. Now, one thing that oftentimes people don't do, and I'm often even kind of guilty of this, the first kind of run through when I do some statistical analyses, is we need to remind ourselves that what we're dealing with is a general linear model. Actually, this is a general linear mixed model, but it's still a general linear model of sorts, meaning, again, about the adding. Parameters are only added and subtracted from one another. They never multiply by one another. They never serve as an exponent. But a really important assumption of this is that our error variance is normally distributed. And so now would be a really good time to check for that, because we know that the raw data going in, it might not be normal at all, because it could be very bimodal if there's a really strong effect of something. There's no transformation that would make that normal, and you wouldn't necessarily want to make that normal. But now that we've run the model, we'd like to look at whether or not that assumption that we made, that the residual variation is going to be normally distributed, is indeed met. And so I'm going to first of all do the old noggin test, which is, let's look at our data. So let's just do a histogram, a hist of our resid. If you recall, that resid function will draw the residuals out of the model, so it's all the leftover variation. Going to look at that histogram. You can see it's somewhat, it looks skewed, and so maybe we're not terribly happy with that. We could also, library fbase6, if we wanted to go that extra step, we could use the normal test function from the fbase6 package and give it the residuals of our mod. And let's go ahead and let's use the DA test, because Jim likes the DA test, and it gives us a little bit more information about where deviations of normality might fall. And so we look at the results from that, And it looks like kurtosis is not a problem. And again, recall that kurtosis is just a description of whether or not you have a lot of mass near the center of your distribution, or whether or not it's more distributed near the tails, which it also should not be. So kurtosis looks fine, skewness looks way off, and then our omnibus test is telling us that overall these do not appear to be normal. And so again, if we looked at our histogram, and then we could go ahead and let's look at our raw data. Let's just do that because that might be curious to us. So I'm going to do that, and then it's leaf.glucose. And so even looking at that, I think I figured out how to make those warnings turn off. We're going to see if this works. Okay. Even looking at our raw data, you can see that the data are clearly skewed to the right. And so what we might be interested in knowing is, if we were to look at that, well, what would our data look like if we had log transformed it? And if we had log transformed it, it looks a little normal. It almost looks a little kurtotic over here and maybe a little weirdly screwed. But let's go ahead and take a look. How does our model do if, let's redo our model. And this time for leaf glucose, I'm going to take the log of leaf glucose in the model. And now I want to look at the residuals of that model. That looks a little bit more normalish. And let's go ahead and do the residuals on that model. Oh, and it looks like it made everything happy and friendly. Look at how happy it is. Yeah. And so that is the important assumption of the model. And I'm just saying that to remind you all that that is what the assumption is. It's not that your data are normal. And I'm kind of curious. I don't even know if our raw data are normal. No, that didn't work. My dev off didn't work. Let's see here. Whoa. Oh, there's the problem. There's the problem. Copy. Okay. Yeah, so even log transforming our raw data does not make our data normal. But that's not the important assumption of our model. The important assumption of our model is that after we remove all the variation from our fixed effects and control for the unknown components of the variation from our random effects, or just in the residuals in general, that that the residuals of the model are normally distributed. And we feel pretty good that indeed they are. So resid the mod, and there we have it. Okay, so we feel good. We know that we want our response variable to be the log transformed of the amount of glucose. And so now we just have to remember when we're communicating our results, or when we're making plots, or we're describing our results to people, that we're explicitly going to be talking about the log of the amount of leaf glucose. So we have our model. We've done the stepwise selection procedure. We can uniquely estimate all the components of our error structure. We feel good about that. Here again is our mod. And so now let us go ahead and run the ANOVA on that. So if I use lowercase ANOVA, I will use the Elmer test function, which overwrites the lowercase ANOVA. And that does it more of the traditional way, using an analysis of variance where it corrects for the number of degrees of freedom in the denominator. And we go ahead and we do that. And what we see is that there is a significant interaction term. And what that significant interaction term is telling us is that we cannot trust the hypothesis testing on our main effects. So even though neonics look like they're significant and snails look like they're not significant, we cannot trust that because we have this significant interaction term. Okay, so all the same rules apply that applied before. We can also use, there's a number of different ways you can do hypothesis testing on these mixed models. The other way would be to use the capital ANOVA model, which is going to give me an error unless I type in library car. Okay, so now I've loaded car in, and I do that, and it does that modification of a likelihood ratio test called a Wald chi-squared test. And using that, we similarly see there is a significant interaction term between neonics and snail. Note that also now snail is popping up as being significant in this model, but again, that's two different approaches to doing the hypothesis testing. And also, we can't trust this anyways because this is what's happening. Okay, so now that that happens, or that happened, we need to split up our model if we want to understand where the statistical significance lies. Again, if we were to look at our model though, just because we're not happy with our hypothesis testing, we are totally happy with these parameter estimates. Based upon those parameter estimates and how the dummy coding works with the toggles, ons and offs, we can come up with a predicted value for any particular combination of either being neonic or no neonic, or snail versus no snail. So I've already typed in a lot of this. One, because I've already done this once and the sound didn't work, but also because it would have been a lot of typing and I would be prone to clearly make a mistake. And so I'm going to just go ahead and my disk space isn't, you don't know what you're talking about. And so what do we have? We're going to split models by neonic treatment. And so we're going to start with ones that were, the neonics were in the treatment group. Okay? And so what we've done here is I'm using the which function, and that's going to tell me which index do the neonics absolutely equal treatment, right? So if we were to just look at the neonics, right, they're either a control or a treatment. I'm asking which ones are exactly treatment. It's that one. Let me try. Okay, I'm back. A little technical issue. It occurred to me that because I had, it occurred to me that maybe my disk was getting full, so I had to do a little maintenance. Okay, so where was I? Right. First we're gonna split the model based upon what neonic treatment you're in. You're either in an experiment that had the neonics added, or you're in an experiment that didn't have the neonics added. This which function here will tell me which of the neonics are in the treatment group, and it's going to give me this list of numbers again right here. And I'm going to use that as an index, so that way I'm going to be telling it that I want to save the second row of our data frame, the fourth row of our data frame, the sixth row of our data frame, etc., etc. And I'm going to do that by simply using that as the index for the rows. And so now I've created this new data, which is neonic treatment for glucose, so I'm calling it neotreat glucose. And I'm going to go ahead and I'm going to throw it back into a new model. Now the only difference between this model and the previous model is that we're not including neonics in this model because there's no difference. There is no factor level in this particular model that is neonics, because the only data that are going to be analyzed in this model are the ones where the neonics were added, the treatment group. And so I'll go ahead and I'll run that, and I can look at that and see what it looks like. I got some new parameter estimates. And so let's go ahead and let's do an ANOVA on that. Copy, ANOVA. Okay, and what do we end up with? Our statistical inference is that there is absolutely no effect of snails when neonics are present. And we could do it also using the capital A ANOVA function. Capital A ANOVA function. And again, no effect of that. Okay? So let's do the exact same thing now for the ones where there is no neonic. And so all I did for here is that I'm creating a new object, a new data frame called neocontrol glucose, whereas this one was neotreat glucose. This is neocontrol glucose. And the only difference is I'm putting that little minus sign in front of that index that we made using the which function. And what that little minus sign is telling it is that I want all of data glucose except for these rows. So it's the exact opposite rows that were in the previous model that we made. And I can go ahead and I can run that, run the ANOVA on that, and it looks like it's significant. Okay, here we have it. So we would accept or reject the null hypothesis, accept the alternative that snails are important for predicting leaf glucose. And likewise, using the Wald chi-squared test, we would arrive at the exact same statistical inference right there. Okay, and so that explains a bit as to why we had that significant interaction term. When there was neonics added, snails didn't matter. When there was no snails added, or when there was no neonics added, suddenly the presence or absence of snails were important. So that explains why we had that interaction term, which again is why it's important that we always have to split up the model to do the hypothesis testing, because we would have been wrong had we said that there was no effective snail. Turns out there is an effective snail, but the only time that we have that effective snail is when we are in the control group. Okay, and so we can similarly do the same thing with split the models by snail exclusion. Okay, so here I'm going to say which one of those snails, right? So here's all the snails. Snails have two levels. They're either excluded or they're not excluded. And I'm going to say which ones are super duper equal to excluded. That's going to give me my index. So these are the row numbers that I want to hold on to. So I can go ahead and I'll do that. Let's copy and paste and make ourselves happy. Okay. And so now I'm creating a new data frame out of the old one, except I'm only keeping the ones where snails are excluded. So there we have that. And now we can go ahead and we can set up our model. And so again, the model looks very similar to the other one, except now snail is not a factor in the model. That's because we're only analyzing the data where snails were not present. So it wouldn't make sense to try to put snails in there as a factor, because you would have 0 degrees of freedom. You only have one treatment level. And so I'm going to go ahead and I will run that. Okay. Oops. And then let's go ahead and say ANOVA on mod.snail.exclude. And there we have it. Significant. All right. And then I bet we get the same answer here. Yep. And so the presence of neonics is important when snails are not present. And so let's do the exact same thing when snails are present. So the only thing here, I'm putting that minus sign again, so it's going to drop those rows. It's going to drop all those rows. So now I'm only analyzing the data where snails were present. And I'll go ahead and I'll run that into my mixed model. And then I can say what's the ANOVA on that. And then the capital ANOVA on that. Okay. Both approaches give me pretty much the same answer. I'm going to reject the null and accept the alternative hypothesis that indeed we should be including snail in the, or I'm sorry, we should be including neonics in the model. So under both scenarios, neonics are always important. Under the model where when we split it up between the neonics, snails are only important when neonics are, is it absent? Let's look. Yeah, so when neonics were added, there was no effect of snails, and when neonics were not added, then there was an effect of snails. And that's what was explaining our interaction term when we ran it on the full model. Okay, and that's why we couldn't interpret that hypothesis testing, because that would be wrong. Because we know that snails actually are important. but they're only important under certain situations, and we can't detect an effect of their importance under different situations. Yeah, and then we still believe these parameter estimates, they're all fine. Okay, that is it. I guess a few things that I'll point out as I was going through this, and obviously the nice thing I suppose about YouTube is that you can hit stop and go back. If we went back to our original full model here, we were allowing there to be a different intercept for snail and plot. When I ran it, notice that I dropped out the slope term here when I split them in half, when I split them apart. And so when I ran that one, it was all fine right there. But if I ran that full model with the intercept term, and again, this is just the reduced data set. This is only the data set where neonics were not added. I got the singular fit warning, and I did that, and there you go. We can't uniquely estimate it, and so I put a 1 there, and so now I do a 1, and there we get the same answer as we got before. It's always a good idea to try to, if you're going to start adding structure to your error, to make it as similar as possible to the full model as much as you can. But it's by no means necessary whatsoever. And again, it's your model. You can do whatever you want with your model. Having that correlated error structure isn't going to really affect the hypothesis testing all that much, but it's something to be aware of. And I will add some annotations to the notes that I'm going to post right now, or that I'll post shortly. I guess it'll be posted before you see this, though. So time, right? It's weird. To kind of explain what those steps were that went along with that. And I believe that is about all. So that was our review of mixed models and thinking about mixed models. Hopefully it was some help as you work on this week's homework, and starting next time I truly will start generalized linear models. But for now, thank you and good night.